Hello everyone and welcome back to AP Bio with AP Playground. Today we're reviewing Unit 6, Gene Expression and Regulation. What you need to know for this unit, first of all, the differences between DNA and RNA, DNA processes and gene expression processes including replication, transcription, and translation, how gene expression itself is regulated and how it contributes to cell specialization, the effects of mutations on genetic diversity and gene expression, and lastly, different methods of biotechnology. Let's begin. Beginning with discussing DNA and RNA, it's important to remember that DNA and RNA store and transmit genetic information. And they are made up of nucleotides, that is the monomer, the building block. And each nucleotide consists of a 5-carbon sugar, deoxyribose for DNA and ribose for RNA, a phosphate group, which gives these nucleic acids a slightly negative charge, and also a nitrogenous base. And DNA has 4 different kinds of nitrogenous bases: thymine, cytosine, adenine, or guanine, often called by just their first letter, T, C, A, and G. RNA has very similar nitrogenous bases, except thymine is replaced with uracil, or U. And some key points about DNA and RNA structure include the following. DNA has 2 strands. It's in the shape of a double helix, as you can see here. And so these strands are anti-parallel, meaning that they are parallel but they run in opposite directions. And the base pairs are going to be A, T, and C, G. Now what I mean by this is when we have our 2 strands, individual strands, each nucleotide is bound to other nucleotides through a covalent bond within an individual strand. But how the 2 strands are bound together are through hydrogen bonds between the nitrogenous bases. And the nitrogenous bases can only bind adenine to thymine and cytosine to guanine. Meanwhile, RNA is a single strand, and this is because it's made for temporary use. And it has a ribose sugar rather than deoxyribose sugar, and the bases are AUCG because RNA does not have thymine, it has uracil instead. So the base pairing also changes up a little bit. Now going past the monomer level, DNA as a unit wraps around histone proteins, which forms nucleosomes, which in turn make up chromatin, and when chromatin is condensed, it becomes chromosomes, like the common shape that you would associate with a chromosome. So when we have DNA wrapped around histone proteins, that is the whole genetic information, that is the chromatin, which can then be condensed into chromosomes. And each chromosome is made up of introns and exons, which are coding and non-coding regions. And introns are going to be non-coding regions, while exons actually have code that contains genetic information that can be expressed. And when we talk about the different types of chromosomes, there are linear and circular chromosomes. And eukaryotes and animals, humans, they have linear chromosomes, like you can see here. Whereas prokaryotes, such as bacteria, have circular chromosome that is only 2 strands of DNA. Lastly, we got to talk about plasmids, which are small circular pieces of DNA in prokaryotes that are extra chromosomal, meaning they don't come from the main circular chromosome of the prokaryote. And so what these plasmids can do is they can be incorporated into the overall bacterial DNA, and they can introduce genes into that DNA. And we'll discuss the uses of this further on in the video. Now we have to talk about DNA replication, and this is when, as in the name, DNA replicates itself, it duplicates itself. And this is a key part of the central dogma, which talks about the process of DNA being converted into outward expression, so into proteins. And DNA replication ensures continuity of hereditary information. DNA makes sure that all of the cells within your body are going to have the same DNA because of replication. And it is a semi-conservative process, meaning each of the daughter molecules has one old strand and one new strand. So what we see here is our original double helix of DNA that consists of 2 strands, and when these are pulled apart, the 2 new DNA molecules that have been created, one of them is going to have one of the strands from the original 2 strands, and the other one is going to have the other strand from the original 2 strands. So each of the daughter molecules has one of the original DNA strands and one new strand. And we'll talk about how the new strand occurs in a second. But DNA replication starts with the helicase enzyme, which unwinds the nitrogenous bases. You can even say unzips by destabilizing the hydrogen bonds between them. And then the topoisomerase enzyme contributes to further unwinding, cuts the backbone, and helps release any tightening as a result of helicase enzymes work. So step 1 is to unwind and separate the 2 strands of DNA from each other by cleaving the hydrogen bonds using helicase and topoisomerase enzymes. Next up is going to be DNA polymerase, another enzyme that binds free nucleotides, so nucleotides that are not part of a whole nucleic acid, not part of DNA or RNA, but just nucleotides that are single and free, and binds them according to our base pairs, our A, T, and CG base pairs, on to the ends of the strands. And it's only going to bind to the 3' end. And what I mean by this is if you're looking at the carbons within a nucleotide, you can count starting from the oxygen molecule of carbons, and you'll see that one strand is going to run from the 5' to the 3' carbon, and the other strand will also run from the 5' to the 3' carbon, but in the other direction. And so the nucleotides are added onto the 3' carbon end, and RNA primers are required in order to begin replication. So DNA replication runs continuously along the 5' to 3' strand, so the strand that follows the replication fork, otherwise known as where the helicase enzyme is unwinding. So for instance, over here we can see here's our leading strand, here's our replication fork, and helicase is unwinding the enzymes. It's going in this direction, and so the strand that runs in this direction from the 5' to 3' carbon, DNA polymerase has no issue with running continuously and base pairing free nucleotides. However, to add nucleotides to the other strand, to the strand that it's running in 5' to 3', but it's running in the opposite direction, it must be copied in segments. And these are known as Okazaki fragments. So Okazaki fragments are formed on the lagging strand, which are then sealed together by ligase. And the last step is telomeres added to the tips of the chromosomes that have been made from DNA replication in order to protect the genes, because every time a cycle of mitosis occurs, there's going to be the ends of the DNA removed, and so telomeres allow for all of the coding regions of the DNA to be protected. So now we begin to discuss gene expression, and this is the process by which DNA directs the synthesis of proteins. So how does the genetic information then result in outward appearance or certain function? And so each gene codes for one polypeptide, for one protein. And the genetic code is nearly universal. There's lots of similarities between how gene expression works across organisms, and also similarities in DNA itself between different species and organisms, which all show evidence for common ancestry. And here we're going to be following the central dogma, which I brought up earlier. DNA is going to be replicated, transcription turns DNA into RNA, and RNA through translation becomes a protein. And so we're going to begin by talking about transcription, which is the synthesis of RNA using DNA as the template. And this happens in the nucleus, whereas in prokaryotes it's going to happen, within the cell, but there's no nucleus there, and that's going to produce mRNA or messenger RNA, which is then sent to ribosomes to undergo translation, which is production of a protein chain using the mRNA transcript. And transcription and translation are going to occur at the same time in prokaryotes. So now talking about the steps of transcription, one strand of the DNA is transcribed. This is the template strand. This is the strand of DNA that the mRNA is going to be based off of. And the other strand is the coding strand. It's going to have the same nucleotide sequence as the mRNA. So what I mean by this is over here, this is our template strand. This is our strand that mRNA is being formed off of by nucleotide base pairing. Of course, with the substitution of uracil for thymine in the base pairing. And the coding strand is over here. It's going to share the exact same nucleotide sequence because both the coding strand and the mRNA are base pairing off of the template strand, essentially. And so they're going to have the same nucleotide sequence, save for thymine in the DNA strand versus uracil in the mRNA strand. And the enzyme that does all of this is RNA polymerase. It separates the two strands and connects free RNA nucleotides as they base pair, adding them to the 3' end of the DNA, and the DNA is read 3' to 5' so that the RNA can become anti-parallel and run 5' to 3'. And so the RNA polymerase is going to attach at the promoter DNA sequence and detach at the terminator DNA sequence because only a certain amount of the DNA needs to be transcribed for certain purposes. And transcription factors assist the process, and we'll talk about those more when we review 6.5. But essentially what's happening here is we're building a strand of mRNA using nucleotide base pairs based off of one of the strands of DNA in order to then export this mRNA out of the cell. But for this RNA strand to become mRNA, it needs to be processed. And processing is only going to occur in eukaryotic cells. Processing does not occur in prokaryotic cells because the mRNA is immediately sent to translation from transcription. So there's going to be a 5' cap added, so a modified guanine nucleotide attached to the 5' end of the mRNA, a poly A tail, which is a bunch of adenine nucleotides attached to the 3' end to the end of the RNA, and these are going to help mRNA export from the nucleus because it needs to get to the ribosome, protect it from degradation, and that's specifically what the poly A tail is for, because by having these extra nucleotides, if some get cut off, and we'll talk about that later, then the coding region of the RNA can still be safe. And lastly, they'll help attach to the ribosome. Now, there's also RNA splicing, where specific sections of the RNA are removed. And just like we discussed with chromosomes, there are introns and exons, and the introns are the known coding regions. And so RNA splicing is going to remove the introns, and then the exons are bound together by a spliceosome. And this happens through small RNAs, such as the ribosome, which is a catalytic RNA, which is pretty cool because most enzymes, most catalysts of reactions happen to be proteins, but a ribozyme is an RNA that fulfills the same purpose. And the coolest part is that an intron in one RNA molecule can be an exon in the other. So from the same primary RNA, you can have different finished mRNA, which will then result in different protein expression because what counts as encoding and non-coding region can change. And so now we're going to move into translation, the next step. And to talk about translation, we need to know two other types of RNA. First up is tRNA, and tRNA transfers amino acids from the cytoplasm to the ribosome to assemble a polypeptide. So it gets the correct amino acids based off of the mRNA in order to assemble the protein. And within the ribosome, which is made up of RNA and proteins, mRNA is going to be read in codons, which are groups of 3 nucleotides. And so tRNA has an anticodon, which allows it to base pair to the codon on the mRNA and get the right amino acid. So a group of 3 nucleotides, one codon, is going to code for a single amino acid. And so ribosomes are going to have 3 sites for tRNA molecules. The P site that holds the growing polypeptide chain, that holds the growing protein, the A site that's going to introduce new amino acids and bind to the anticodons, or bind to the codon using the anticodon, and then lastly the E site where the tRNA that has deposited its amino acid will exit so that it can then go get another amino acid and participate in the process. So if we're going to practice going through transcription and translation, the question is, what amino acid will a DNA sequence of GTA code for? So first we have to convert this into mRNA. So we have to do some base pairing. GTA is going to become CAU. Guanine, cytosine, thymine, adenine, and adenine in DNA would be thymine, but in RNA it's uracil. But then we have to find the anticodon. So we have to do base pairing one more time to get GUA. And once we've gotten our GUA, that's our anticodon, that's our tRNA molecule, and now we can find what amino acid that codes for using the codon chart located over here. So if we look at our first letter, that's G, so that's going to be right over here in this row. Then our second letter is U, so that's the first column, and our third letter is A, so third row. GUA codes for the amino acid valine. Now, specifically how these three sites within the ribosome work and the steps of translation, the small ribosomal subunit, because there are two ribosomal subunits that make up a ribosome, we have the large and the small. So the small subunit binds to the mRNA, so the start codon, AUG, is placed in the right position. And the codon AUG is going to code for methionine. And then the first tRNA is going to bond and the large subunit attaches. Then the tRNA with correct anticodon is going to bind to the A site where tRNA enters, and the peptide bond is now formed. And what's going to happen is the amino acid in the P site is going to move over to the A site, and then all three are going to shift over, or in this case two, because it's the first step. So the first tRNA molecule is going to exit through the exit site. The one that brought the second one is now going to be holding the growing polypeptide chain, and now a third tRNA molecule can enter. So what's going to happen is the polypeptide chain will be shifted over one tRNA molecule, and then they will all be moved down one step within the ribosome in order to make space for the next tRNA molecule. And once a stop codon in the mRNA is reached, and there are three options for stop codons, and this codes in case of mutations, we'll talk about later, but there's redundancy within the codon chart. The polypeptide becomes freed, undergoes protein folding, and it's going to have a signal. So a signal peptide is a sequence of 20 amino acids that determine the protein destination. So this protein folding of primary, secondary, tertiary, quaternary structure and the signal peptide are going to determine then what the protein is going to do within the cell, or if it's going to be exported out of the cell. And so far we've been discussing the central dogma in the stages of DNA to RNA to protein, but we also have to discuss retroviruses and an alternate flow of genetic information. So retroviruses, or viruses, can only quote-unquote reproduce, because there are debates on whether they're living or not, but they can only replicate by infecting a host cell, and they have genetic information stored as RNA. So when they infect the host cell, reverse transcriptase and reverse transcription occurs, where the RNA is first turned into DNA, then it can enter the DNA of the host within the nucleus, and then it'll be transcribed and translated in order to produce new viruses. Now we're going to move on into the different ways that gene expression can be regulated both within eukaryotic and prokaryotic cells. And there's a lot of ways that this can happen in eukaryotes, and we'll start with epigenetic changes such as DNA packing. So every cell in an organism has the exact same genome, but not every gene is expressed in every cell. So within your body, every single one of your cells, they all have the same DNA because they all came from one cell that underwent mitosis, and you all have the same within your cells the same DNA, but not every gene is expressed in every cell. You have cells that have different functions and specific purposes, and so gene expression needs to be regulated, and this can happen before and after transcription and before and after translation. And an epigenetic change is when the DNA sequence is not changed, but its expression is affected. And this happens in eukaryotes through DNA packing. And this just means that the more tightly bound the DNA is around the histone proteins forming that nucleosome, the less accessible that DNA is for transcription, so the less chance that this DNA will be transcribed. And so DNA methylation adds methyl groups and causes tighter packing of the DNA, while histone acetylation adds acetyl groups, which then result in less tightly packed DNA, which is now more accessible for gene transcription. And again, this is a eukaryotic method. So now that the gene is available, the DNA is available for transcription, let's talk about how transcription can be regulated. And again, this is eukaryotic. So we have regulatory sequences, which are stretches of DNA that interact with regulatory proteins to control gene expression. And some genes are always on, they're constitutive, while others have inducible expression. So in order to express the gene, certain factors need to be present. It needs to be an induced expression. And so regulatory sequences can promote expression or block expression. So we can have inducers that promote expression of the gene or repressors that are going to block it. And so we have a transcription initiation complex. And when an activator protein binds to an enhancer and general transcription factors are involved, the DNA is going to bend over, bringing this complex to the promoter, and that's going to promote the attachment of RNA polymerase to the promoter and starting gene transcription. So all of these transcription factors and activator proteins come and play together in order to regulate transcription. And transcription factors can control multiple genes at one time in order to coordinate an expression of genes. Now, we discussed this when we talked about mRNA processing, but after transcription, this alternative RNA splicing, meaning that the same mRNA strand can have different introns and exons, different coding and non-coding regions, this will change each strand of mRNA, and that depends on the goal of gene expression, depending on what genes need to be expressed at that time. And having a longer poly-A tail, like we discussed, will allow the mRNA to survive longer. And it now has protection from exonucleases, which are enzymes that are going to be looking for discarded nucleotides in order so that they can become free and used within DNA replication or within transcription, and will cut off nucleotides off of mRNA, but the poly-A tail allows mRNA to survive these exonucleases. Now, regulatory proteins can also block translation or promote translation, just like we discussed for transcription. And after translation, protein activation can be inhibited because, of course, in order to activate the protein, folding must occur. So we have to achieve that secondary, tertiary, and potentially quaternary structure for that protein in order for it to be functional. So if the folding is inhibited, then the protein will no longer be functional. Additionally, even after the proteins have been folded and activated, they can be tagged with a substance known as ubiquitin, and that's like a signal that the protein needs to be degraded. So there are so many steps that can regulate gene expression within eukaryotes, and these range in all of the stages of the central dogma, leading up to even after the protein has been created. Now let's take a look at gene expression and regulation within prokaryotes. So within prokaryotes, gene expression is regulated through the use of operons. And operons are genes clustered into groups, and they're controlled as one unit, sort of like transcription factors inducing transcription for multiple genes in a coordinated effort. And operons consist of the group of genes that can be expressed, the operator, which controls the access of RNA polymerase to the gene, and the promoter, which is where the RNA polymerase attaches. And the operator is able to have a repressor bound. And when the repressor is bound to the operator, RNA polymerase is not able to access the genes and induce transcription. And there are two kinds of operons. There are inducible and repressible operons. So one example of an inducible operon is the lac operon. And it contains genes for digesting lactose. And it's inducible because it's normally off, so it needs to be turned on. So when the operon is off, that means the repressor is on, as you can see here. We have the repressor preventing the RNA polymerase from expressing these genes, and it is a catabolic process. So lactose digestion enzymes are only going to be produced when they're needed. So we only need to break down this material when it's present within the cell. So in a normal state, the operon, the lac operon, is going to be off. However, when lactose becomes present, there is now an inducer, such as allolactose, which can bind to the repressor protein and deactivate it, allowing the operator and the RNA polymerase to function, and now these genes can be expressed for digesting the lactose. So it's only going to turn on when there is a certain substance present in the cell. Now, the other type of operon is a repressible operon. It's going to be innately on, so there's not going to be a repressor, and generally these are production operons. They're going to be making something, such as the tryptophan or TRP operon. And so tryptophan is going to continuously be produced until there's too much. And when there's too much tryptophan, it'll bind to the repressor, activating it, turning the operon off. And this is sort of like feedback regulation. So the operon is going to be working until there's too much of the product, and then it will turn off, and these groups of genes that are producing tryptophan will no longer be functional. Now, what is the point of all of this regulation of gene expression? Well, the phenotype of a cell or organism is determined by the combination of genes that are expressed and the levels at which they are expressed. And of course, the phenotype is going to be the outward appearance of the organism. So the function and the amount of the proteins that are created as a result of gene expression are going to result in the outward appearance. And transcription is going to begin during development, at the very beginning, and gene expression will continuously occur throughout development. And so cell differentiation results from the expression of genes for tissue-specific proteins. And what I mean by this is when different genes are expressed within different cells, then these cells are going to become different. They're going to have different jobs, even if they started from stem cells that have no innate function. But within eukaryotes, within us, we have all these different kinds of cells with specific functions: intestinal cells, liver cells, cardiac cells, nerve cells, blood cells, muscle cells, and these all occur from regulation of gene expression. So we have differential gene expression, the expression of different genes by cells with the same genome, and that's going to influence the cell products and the cell functions. Now, one regulatory molecule that we haven't discussed for gene expression are small RNAs, such as miRNAs and small interfering siRNAs that can bind to mRNA and then either degrade the mRNA or block its translation. So these also can interfere with mRNA after transcription, further regulating gene expression. Now it's time to talk mutations. And a mutation is a random alteration in DNA sequence. So a change in the nucleotide sequence of DNA that happens randomly. And it can affect changes in the amount or type of a protein produced, altering the phenotype because of point mutations, which are alterations of just one nucleotide base pair. But even one nucleotide base pair can have a large effect. If a nucleotide is substituted for another, then the codon is different. If there's an insertion or deletion where there's an addition or loss of a nucleotide pair, for instance here, where we have a normal sequence for 3 distinct codons that are going to code for 3 proteins. If this insertion occurs, the new codon is now ATT, and this new codon is now GCA, and this new codon is now TTG. So now we can be coding for 3 absolutely different proteins once we go through transcription and translation. And this is known as a frame shift mutation. It shifts the frame of 3 codon groupings because the groups of 3 remain, but now what's in them are different. And that's going to be a mutation that can really change the type of a protein produced. Now, nonsense mutations cause premature stop codons. And we discussed that there are 3 different stop codons. You can see over here, it's UAA, UAG, and UGA. And this is done in case there's a mutation that changes a stop codon so that the stop codon remains, but a nonsense mutation can cause a premature stop codon, so stop before the polypeptide is complete. However, there can also be silent mutations with no effect on the resulting protein. For instance, if you take a look at the tRNA needed to create valine, you can see that there are 4 different types of nucleotide bases sequences that are able to create valine, and they all have the same first 2 letters, but the 3rd letter can be either of the 4 nucleotide bases, and this redundancy allows for protection within the expression of the code even if a mutation occurs, and that's what we call a silent mutation. And mutations are caused by mutagens, which are substances that interact with DNA in ways that cause mutations, such as radiation, but also errors in DNA replication, errors in DNA repair, errors that happen during the cell cycle. And so an alteration in chromosome structure can also lead to genetic disorders. And here we're talking about non-disjunction, which is when chromosomes do not separate. There's changes in the chromosome number of the cell. And what happens as a result of non-disjunction, where chromosomes don't separate correctly during anaphase, is we end up with cells that have more chromosomes and less chromosomes than are necessary, and that can severely screw up the DNA. But also, if this happens in meiosis and separation doesn't work, then all of a sudden you're going to have, you could get an individual then that has too many or not enough chromosomes born as offspring. And whether mutations are beneficial, detrimental, or neutral depends on the environment. And they're the primary cause of genetic variation. And so when we have a mutation, it doesn't necessarily have to be bad. Mutations are often framed as bad, and they can be bad, but it depends on the environment. Because if a mutation helps an organism survive in a certain environment, then it's going to be passed down as a trait. And for an example here, I have sickle cell anemia, which is a mutation that's going to turn your red blood cells into the shape of a sickle, which can clog blood vessels. But if you're a heterozygote for sickle cell anemia, you have heterozygote trait, then you will have better resistance to malaria. So as a result, this mutation continues to be passed down because it is beneficial to the people living in an environment where mosquitoes and malaria infections are high. Genetic variation also increases in prokaryotes through horizontal gene acquisition. And this is how prokaryotes are able to get more genes into their system. So first of all, we have transformation, where DNA is taken in as a plasmid. And we discuss plasmids as little extra chromosomal pieces of DNA that can then be incorporated into the overall bacterial DNA, into the chromosome. And now these genes are part of the bacteria. Transduction is where viral DNA is taken in. So when the virus infects a bacteriophage, which is a virus that infects bacteria, is when their DNA is taken into the bacteria, then now this DNA is part of the bacteria and can be expressed. And related viruses can even recombine DNA, so DNA can swap if they infect the same host cell at the same time. And of course, a virus is an infectious agent that replicates only inside the living cells of an organism. There's also conjugation, where DNA is transferred through cell-cell connection, and those pieces of DNA that are transferred can be incorporated into the chromosome. Lastly, we have transposition, which is movement of DNA segments between DNA molecules. So within the DNA molecule, pieces of DNA can even be moving around. And that's also going to increase genetic variation and introduce different genes into different organisms. For the very last part of our Unit 6 review, we're going to discuss biotechnology, which is the process of manipulating organisms for the purpose of making useful products. And so one of the most impressive acts of biotechnology has been DNA sequencing, which was determining the complete nucleotide sequence of a DNA molecule using base pairing. And we have been able to determine the entire human genome through the Human Genome Project and map the entire nucleotide sequence of the human genome. And this allows for comparison of DNA between species. And some really cool facts can be found from this, such as humans and potatoes share 50% of their genomes. That's how much of our DNA is related to potatoes. And another piece of technology is polymerase chain reaction, which amplifies a piece of DNA without using cells. So it allows replication to happen without cells having to undergo division. So the pieces of DNA, the strands of DNA, are quickly heated in order to denature the hydrogen bonds between the two strands. Then cooling happens and primers attach. And lastly, DNA polymerase is going to add nucleotides to the 3' end of a primer. So it's sort of like DNA replication, but it's happening for a specific piece of DNA, and it's not happening as part of S phase in preparation for division. It's happening just because scientists would like to amplify this piece of DNA and make a lot of copies. And so after the first cycle, we can see that we have now two pieces of DNA that have been replicated, but second cycle goes up to 4, and eventually after 20 to 30 cycles we can have millions of copies of the same exact piece of DNA. Also on the table here is gel electrophoresis, which is a lab technique used to separate DNA. And so DNA is placed in a gel matrix that allows molecules to move through it, and an electric current is applied to the field. And as DNA is negatively charged due to the phosphate groups in its backbone, it'll migrate to the positive end of the field because opposite charges attract. And smaller fragments are going to move the fastest through this gel matrix. And as DNA separates, the difference in banding patterns allows for diagnosis of disease, of determining who the parents are of a person, also helps within crime scenes and matching the DNA found at a crime scene to suspects. As we can see here, we see the crime scene DNA and how the bands are arranged here and how DNA has separated, and we can match that to the DNA of all these suspects, and we can see that the banding patterns of suspect 3 are exactly the same, and they match the banding patterns of the DNA found at the crime scene. And for the very last slide here, we're going to talk about recombinant DNA and gene cloning. So recombinant DNA is artificially made DNA using DNA from different sources. So this is such as introducing human genes into bacteria, which can then be introduced as a plasmid and taken up by the bacteria. As we can see here, this plasmid that could contain human genes is introduced into the bacterial chromosome, and once the bacteria divides and starts expressing its genes, these human genes will be expressed by the bacteria. Additionally, we have gene cloning, where scientists can produce multiple copies of specific pieces of DNA using bacteria by turning the gene into a plasmid and entering it into the bacteria. So even if the bacteria don't take up that plasmid into their chromosome, just through their replication, that plasmid will be duplicated, and now there's going to be more copies of that DNA. We have had gene cloning. And this is all due to the possible due to restriction enzymes such as CRISPR-Cas9, which cut DNA at a specific location. And so we can look here at restriction enzymes that are going to create a cleavage within the DNA, and we have restriction fragments, but one single-stranded end is going to be sticky. So as you can see here, the cut isn't entirely chopping in half, it's just separating the bonds between nitrogenous bases in two of the strands, and so there's going to be a bit of single strand hanging off both pieces of DNA, and those are known as sticky ends. And then they can be bond together through complementary strands using ligase, the enzyme ligase, which will find complementary base pairs that allow for these sticky ends to bind, and then ligase will seal up these sticky ends. So gene cloning is able to happen through restriction enzymes because these enzymes such as CRISPR-Cas9 can cut DNA in specific pieces in order to get you that nucleotide sequence that you want and not more of the DNA around it. So this was a review of Unit 6, and we shall see you in Unit 7.